Everybody doing there today? I hope you guys are doing all right. If we haven't met before, if you didn't manage to make it there last week, my name is Ian Coates and I'm your facilitator for PSWR 1043, and welcome to week 2. Last week there we covered chapter 1 and chapter 2 of the textbook. If you do not have the, sorry, I'm a little out of breath. I've been running around trying to get the kids off to school and all that kind of stuff. And whew, there we go. Take a breath, have a cup of coffee. Last week there we covered chapter 1 and chapter 2 of the textbook. If you don't have the textbook, I highly recommend it. Okay. This week there we're covering chapter 3 and chapter 4. Okay, we're going to look at the cell, and you can see there we're kind of progressing from the minute to the macro, and that's where we're going to be heading soon. I think it's is it week 3 or week 4 there we actually hit our first organ system, which is the integumentary system, the skin. All right, which will move on to bone and muscle and cardiovascular and urinary and digestion and respiration and reproduction and endocrinology. We're hitting it all. So this course is a doozy, all right? And I can't stress it enough that if you don't have the textbook, you should go and get it. Does it have to be the textbook? No. Any introductory biology textbook, you know what I mean? One of those ones there. You get it at a secondhand bookstore. Now, of course, the chapters won't be the same and all these kinds of things, but generally speaking, that's where we are. Why am I saying this? Next week there, we have our first quiz. Now, you'll know there that this course is composed of 2 assignments and 6 quizzes. Okay? Anyone there who's joining late, it's all right. You remember there, you haven't missed anything. We're just yabbering. I'm yabbering on. But our first quiz will open there next week. Okay, and if you go to the, can I do that right here? I'll share. Where are we here? Where's my Word document? Come on. So you'll see here that every week there I include these little pre-test quizzes for the most part, okay? And there are your questions, there are your answers, okay? And that just gives you a little bit of information about the chapter that we are talking about. Where did my syllabus go? Is that it here? Yes. Okay, good. So just going into the syllabus for the course, okay, that's us there. There's the link. Hopefully there everyone is here. If you're here, you obviously found it. What you're really the textbook is right here, okay. Now again, does it have to be the textbook? Does it have to be the brand new 2024 7th edition? No, it can be the 6th edition. I think I've got the 5th edition, and I've checked it all over. Everything is good. Editions really don't change that much per se from edition to edition. Save some money. I know times are tough. This is where we're at, okay. And let us go to here. So this is where we are right in this block right here. Okay. Next week there, after class, you'll see. Next week there, we'll deal with chapter 5 and chapter 6. Okay. But after class finishes there next Wednesday, I will open up quiz 1. Okay, and I don't want you to worry. I know people are working. They got kids and grandparents and mortgages and rent. You know what I mean? I get it. You got one job. Most people have 2 or 3. You know what I mean? That is the way things are, unfortunately, in 2024. But this test there, Number one, I've received a couple of emails there this week, and people are worried because, you know, you look at last week's information. We did intro to the human body, basic chemistry. There's a lot of info in there. Like, this is kind of mind-blowing. I get it. There's a lot of information. You guys have other courses, these things going on, family life, and you know what I mean? I know life is busy. So these quizzes there that will open up. Okay, there's quiz 1, quiz 2, quiz 3, quiz 4, quiz 5, and then the last week there, quiz 6, right? And there's the 2 assignments that are interspersed throughout. We'll talk about those there later. But you'll see there that quiz 1, it's only on weeks 1 and 2. Okay, even though next week there we will be looking at weeks or chapter 5 and chapter 6 there for week 3. Quiz 1 will open up after. Okay, and it'll only be on weeks 1 and 2. The test is completely open book. That's the thing. Okay, so and you will have like the first test there, it's 10%. The second one there is 10%, just to get you going. Because maybe you haven't been in school in a year, 2, 5, or 10 years. I even had some students there haven't been in school in 20 years. Welcome to a community college. Okay, it's a beautiful thing. It's about community, and my classes there, they range from 17-year-olds up to 65. Okay, and everyone's welcome, and I love it. We never stop learning, and don't think you'll ever know everything, because that's impossible. I don't know everything, and life is a journey. Keep learning, right? So your quiz will open up there next Wednesday after class, and it will be open for the week. You'll be able, you can go on Blackboard, you can look at the test, you can start it, stop it, it will save everything, you can change answers, you can go back. The difficult part about an open book test that's open for a week is that you tend to second guess yourself. And that's why these first couple quizzes are only worth 10%. Okay? Learn how to take a test again. All right? That's why, so 10%, 10%, and then you'll see there week 7, quiz 3, they start to jump. All right? Because you've had some experience taking my tests. And my tests generally are multiple choice, okay, true and false. And they're not really, even though these are quizzes, so we call them, they'll be about 50, 60, 70 questions. Okay, they're good ones. So you take your time. It's a journey, all right, and it's your journey. And hopefully there you guys have a chance to meet and greet. I think you guys should be able to message each other there. And if there's any issues, let me know. Okay, and I don't want you guys to. Well, I can't say I don't want you guys to stress, because there's 2 kinds of stress. Okay, there is acute, which is short-term stress. Kelly Clarkson, what doesn't kill you makes you stronger. That is the good stress. These little short bursts of stress there when you're doing tests and assignments, and I've got deadlines there on Friday, I got to get that done. Organization is key. Okay, as well, please make sure that you have a little area that is yours for studying. I do want you to tell me if there is something troubling you, okay, that you can't get your studying done, you can't, if you let me know that there are issues going on in the background, I don't need to know specific details. There are other doors that can open to assist, okay? So basically there we've got quiz 1, 10%, quiz 2, 10%, and then you'll see quiz 3, 13%. The big banana, quiz 4. And you'll see quiz 4 is on week 7, 8, 9. That one there is worth 18%. It's all heart, cardiovascular, blood vessels, these kinds of things. Okay, so I do understand that there's a bit of anxiety there. The amount of information there last week is redonkulous. Well, this week is going to be the same, okay, if not worse. But that's okay. The material doesn't bend. You are the one who is going to bend. And what I mean by bend, you're going to learn how to adapt. You're going to learn how to take these tests. You're going to learn how to increase your study habits. Training this, That's it. You know, the whole idea, teaching an old dog new tricks, right? Well, I'm an old dog. And the learning how to learn again is a skill unto itself, and you will get it. And if there is any issues, you just shoot me an email and we'll work things out. I promise. I'm not in the habit of failing students, and I don't want that happen here. And is this course absolutely doable? Yes, I promise. Okay, you just stick with it. All right, get the textbook. Get the textbook. Read the stuff, and it's also good for as these tests are open book. You're going to have to dig. You're going to have to dig through the PowerPoints. You're going to have to dig through the textbook. Okay, your assignment there goes out week 4, and that one there is all digging. Okay, so just keep at it. Let me know how things are going. All right. And I'm just going to expand this there. I don't know what I just did there. I don't know. I don't know why. It won't. Okay. No. Interesting. But anyway, if you have any questions, just let me know. I'm going to be closing the screen up there so that I can show my screen in the PowerPoints. If you do have any questions, just let me know. You can shout it out. We're all adults here. Just keep on going and keep me posted, okay? Does anyone have any questions at the moment? Okay, let me know. Just shout out. I don't need a thumbs up or anything. Just give me a shout out and away we go, okay? But other than that, we're rocking and rolling. I'm going to try and put that down there. Okay, very good. So, if there are no questions, I can't see if you have your hand up or anything like that. Just give me a shout out. Turn your mic, turn your mic on, and just say, "Hey, I have a question about this particular slide or whatever," and we'll work it from there. Are we good to go? All right. Welcome to Chapter 3, Cells. Okay. Last week was more of an introduction, kind of giving you an overview, a little bit of basic chemistry, where we looked at the atom. Make small molecules. With atoms there, we can make big molecules, what we call macromolecules. Here you're talking protein, DNA, all right? And then we move up into cells, a collection of cells that do the same job. Now we're into tissue, a whole bunch of tissue doing the same thing. Now we get into an organ, and then how the organs interrelate with each other, organ systems. And then we have the human body in all of its magnificence. So, in order to get there, we got to get through cells. So here we go. And again, if you got to leave, you got your kid is sick, or you got to go to work, or you got something else to do, feel free to go. All right? You don't need to tell me. I'm recording all of this, so it will be posted there immediately after class so that you can go back and review it and dig through some stuff. Okay, let's go. If you didn't attend there last week, just remember there that as far as viewing the PowerPoint, this is how I do it. I don't go slideshow because I like to play with my slides, and I get a nice bigger screen to play with. More coffee. So, talking about the cell, labeling it. Explain the role of the nucleus. Identify the components of the cell. And here we're talking about organelles. Us, as human beings, we have organs. All right? Cells have organelles. And these are just little constituents there that they all have their own little job, and they communicate with each other. One organelle is no important than the other organelle. They're all doing their job there for the facilitation and the proliferation of the cell itself. Transport mechanisms inside and outside of the cell. Okay, we are 70% water. Okay, so is a cell. The majority of a cell is water. So you'll have water inside the cell and water outside the cell. So what's the only thing there that doesn't mix with water? Oil or fat. And that's what is the comprises there of the cell membrane. Okay. We're going to look at the phases of the cell cycle. We're going to go into mitosis, not heavily. Okay, when I design this course, like I teach a number of different anatomy and physiology courses. I teach for the biotech, I teach for the pre-health, I teach there for the PSW course here, and it's all about objectives. You know, I try not to go into things there that you aren't going to encounter in your future job as a PSW. Okay, does it mean there that you stop at a PSW? Please no. After you graduate from your PSW course there, go on and be a PSW for a year or two, and then we'll see you back at college, and hopefully there you'll be enrolled in a nursing program, okay, RPN, RN program. There are bridging programs there. Come back, become an RN. Yeah, that's the whole thing, all right. Cell differentiation, we'll look at cell growth, and a beautiful word there, apoptosis. Cell theory. A cell is the structural and functional unit of life. How well the entire organism functions depends on the individual and combined activities of all of its cells. You know, the whole idea there, we are what we eat. Oh, absolutely. Lifestyle plays an important role in our life longevity. Okay, I'm 51 years old. I have friends from college and university back in the day, and the average life expectancy there. You're looking at oh hi, somebody's microphone is on. If you could mute that, unless you have a question, all right. The average life expectancy there, females is somewhere around 88, 87. Males is around 81, 82. Generally speaking, we are what we eat, and lifestyle changes. Your lifestyle constitutes exactly how long you're going to live, generally speaking. And I've had the people are having heart attacks younger. Lifestyle changes. Lifestyle is everything. Young people are getting type 2 diabetes, which type 2 diabetes is a disease that is generally for older people. Lifestyle is everything, okay? Medical practice has to be a preventative, okay? You can't go to the doctor with stage 4, cancer or something like this, and say, "Can you help me?" Because that's the end. You have to start now as far as your health goes. Cell diversity, there's over 200 different types of human cells, okay? Types differ in shape, size, component, structure, function, and here are just some of them here. Here we're talking about cell specification. You'll see what's going on. We have epithelial cells, okay? These are cells there that connect body parts, form lining, transport gases, and when you're talking epithelial cells, things are moving from one side to the other. You'll find epithelial cells there in your lungs. Okay, oxygen, we breathe in oxygen, and that oxygen moves through these guys into our bloodstream. Carbon dioxide, bloodstream, through these cells to the outside world, right? They line our GI tract, our intestinal tract, right? We eat a banana, and it moves into our small intestine there. The nutrients from that banana move from one side to the other. Okay, into our body. So those are epithelial cells. We've got erythrocytes there. Those are a big fancy word for RBCs, red blood cells. Okay, and you'll see their interesting shape. We'll talk to you guys about those there later on. Fibroblasts here, we're talking connective tissue. Okay, here we've got skeletal muscle tissue. Okay, which is voluntary, right? Skeletal muscle. I am in control. But what happens to that banana as soon as you swallow it? You don't see it again for another 36 hours, if you get what I mean, right? So who or what or how did that banana move through the entire digestive tract? Well, there's smooth muscle, and this is involuntary. You do not have control of this. The other kind of muscle, cardiac muscle. Oh, we'll talk about all these there in great detail. Cells there that store nutrients. There's a fat cell. Okay, we got macrophages. These ones here, I call them Pac-Man. Chomp, chomp, chomp. when a bacteria or some type of invader comes on in, chomp, chomp, chomp. Hopefully these bacteria, your body's able to handle them there, and you get the chomp. Okay, our old cells, they get the chomp. All right, so we'll talk more about these there. This one here, the most easiest to recognize of all the cells. This is a nerve cell. Okay, this is a sperm cell. We'll get into reproduction and all these. So you'll see the whole, all these different cells, they all do different things. All cells have common structures and functions. For the most part, human cells have three basic parts. There's your plasma membrane. Okay, flexible outer boundary, usually composed of fat. Okay, because water outside the cell, water inside the cell. It's an oily membrane there that keeps the outside out and the inside in. Cytoplasm. This is what we call an intracellular fluid. Okay, like we have blood to move things around. Well, cells have a cytoplasm. It's like a jelly-like matrix there, and it allows things to go from one side of the cell to the other. And lastly, there is the nucleus. Okay, bacteria don't have a nucleus. They're very primitive organisms there. That's how we know they've been around longer than us. Okay, we are later on in the evolutionary cycle, and we have what's called a nucleus within the cell. It houses the DNA, which is our blueprint. The master control center dictates what goes on in the cell. And to no further ado, let's have a look. There is a beautiful, typical looking human cell. Okay, now we call this bacteria are called prokaryotic. Okay, with a K, prokaryotic. We are what we call eukaryotic. Okay, we're much more highly advanced than a bacterial cell, and you'll see all these different organelles that are going on here. Okay, so here's your cytoplasm, and I'll just zoom right in. Okay, that's that jelly-like matrix there that, you know, keeps everything where it needs to be and allows things to move from side to side if so needed. This is the nucleus. Okay, and this houses the DNA. Okay, there's your nucleolus inside, and this is your chromatin right in here. This is your DNA. You can kind of see it here, these fuzzy little things in that will change according to mitosis. You'll see on the outside there, here is your plasma membrane. Okay, just remember there that the cytoplasm, this is mostly water, and outside of the cell will be water as well. And you'll see there that this membrane here, what we call a phospholipid bilayer, okay, it's just composed of a fat, an oil, all right, because water and oil don't mix. So if you want to make 100 trillion little apartment buildings in our body and call them cells, we are mostly water. Keep the water out, keep the water in using an oil membrane. There's lots of other things going on here. Just move this over just a little tad. Perfect. And you'll see there that we have ribosomes. Oh, we'll hear about these ones, okay? There's rough endoplasmic reticulum. And you'll see it has these little ribosomes all within it. Okay? So this endoplasmic reticulum and rough has these little ribosomes all within them. Okay? Ribosomes make protein, which is very, very important. Your body can store sugar. Your body can store fat. Your body can't store protein. And you look, your hair is growing every day, nails are growing, you know, all these different, you know, eyebrows, eyelashes, all of our connective tissue there connects our bones together. And, you know, we need a constant supply of protein. Ribosomes make protein. I'd write that one down. Yeah. What else we got here? Just kind of, I'm going to zoom out a bit here. You've got microtubules. You see there, they actually hold, here I'll move so I can get some hands going in here. These cytoskeleton, it holds the cell. You see the cell has this shape. Well, why does it have this shape? Because of these cytoskeleton microtubules. Okay, they're holding the cell out. These ones here, mucho bueno importante. These are what we call mitochondria. These are the powerhouse of the cell. The whole idea there that you eat a banana and you extract the energy from it. Well, how does that happen? These are the guys that do it. Mitochondria. All right, we'll get there. There's centrioles, lysosomes there. These ones here are the garbage people. They, while the body is perfect, there are little bits and bobs of protein and fat and this and that left around. These guys here, they degrade them. All right. There's smooth endoplasmic reticulum as opposed to rough. And you'll see there, these guys here do not have those ribosomes. And they're involved in packaging. All right. And you'll see there, the Golgi apparatus as well is involved in this here. And you'll see there this packaging. If something needs to get exported outside of the cell, that's how it goes. We'll get into all these there. Lots of fun, lots of fun. Let's go. So extracellular material, these are substances that are found outside of the cell as opposed to intracellular material. Okay? So there's classes of extracellular material include body fluids, interstitial fluid. Okay? This is that's the fluid there that is in between all of our cells. Cell, 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 cell. The fluid that is outside in between these different cells, we call this interstitial fluid. Blood plasma, fluid of the blood. And when we look at the constituents of blood, I know blood just looks like a red liquid, okay, but it's not. Well, it is. But there's lots to the blood. There's RBCs, there's white blood cells, WBCs, there's all these other things going on. And you know, like if you get a cut and you get this yellowish fluid oozing from the cut, that's your blood plasma, okay? CSF, cerebral spinal fluid, okay? The whole idea there that if you go to these Zehrs or No Frills or whatever and you go and buy some tomatoes, well, these tomatoes there in the wintertime, they came from California on a truck and they're bumping on the truck the whole way from California. Well, you look at the bottom of the box, those tomatoes there, the bottom of them are all bruised because they're getting whacked, right? And why do you think our brain is sitting on a fluid? The same reason there that if a person who breaks their leg, they can go into a swimming pool and get some exercise because of buoyancy. The water is holding up us. Does the same thing there with the brain, so the bottom of our brain doesn't get bruised. Good one. Cerebral spinal fluid. Cellular secretions, saliva, mucus. Okay, extracellular matrix there. Substances that act as glue to hold the cells together. Cytoplasm is a gel-like substance found inside the cell, okay, but outside of the nucleus. Okay, no cytoplasm within the nucleus. Okay, it's composed of cytosol. There's your gel-like, primarily water, electrolytes. Electrolytes being salt, sodium, chloride, potassium, these kinds of things. Those are your electrolytes. Okay, remember those ions that we talked about there last week, right? Okay, that's what these electrolytes are. The same thing there that's found in a bottle of Gatorade. Don't buy Gatorade. It's a big scam. Nutrients as well, and organelles. We call these little organs. The plasma cell membrane acts as an active barrier separating intracellular fluid, ICF there, from extracellular fluid, ECF. Controls what enters and what leaves the cell. Okay, and that's the whole point of this membrane. It's selectively permeable. Some things can just readily pass through, some things cannot. They need help to get through. And it's all about, you're going to see a word called diffusion. The whole idea there that things move from a high concentration to a low concentration. And you think about that. If I take some perfume and just go and in the corner of a room, somebody on the other side of the room immediately can't smell that. But within about 5 minutes, it's very high concentration of perfume in the corner, low concentration over there, but within about 5 minutes, things move from a high to a low naturally, and all of a sudden, in about 5 minutes there, the whole room is going to smell like perfume. Okay? We'll talk more about that as we go. Specialized membrane proteins float through this fluid membrane, resulting in a constantly changing pattern. We'll call this there a fluid mosaic. We'll talk more about that there in a bit. Surface contains what we call these glycoproteins. Glyco means sugar. So these sugar proteins on the outside of a cell. I'll show you these. This is how our body differentiates from our cells to a foreign invader. Everybody has their own specialized glycoproteins on their cells. And this is how our white blood cells differentiate our cells from someone else. And if we have and like someone there who gets a blood transfusion, someone there who gets an organ donated from somebody else, okay, they won't have the same glycoproteins on these cells as we do. So that's why someone there who gets an organ, they have to take immunosuppressing drugs for the rest of their life, because the body is going to keep attacking that new organ. Membrane structures help to hold cells together through what we call cell junctions. And this is just like a typical cell membrane right in here. Okay? And you'll see that these are these glycoproteins. Okay? Right in here. There's your carbohydrate chains, and this is what our white blood cells look at. I call them the sniffer dogs, these white blood cells. They're always watching. They're always looking for foreign cells there. Okay? And they see our cells, they recognize our cell pattern. Okay, that one's good. All right, yep, this one's good. And if they recognize there a glycoprotein chain there that is not from, not recognizable, then all of a sudden, the alarm bells sound. We have a foreign cell. Okay? You'll just zooming in here. If this is extracellular space outside the cell, intracellular space, there's your cytoplasm inside. And you'll see here that this is water in here, and you'll see that this is what we call a phospholipid bilayer. And you'll see there that these hydrophilic heads like water. Hydrophilic loves water. Hydrophilic loves water. And you'll see these hydrophobic, water-hating tails on the inside. This is the fat component there of the cell membrane. Now, is it just like a piece of cellophane or plastic wrap that circumvents a cell? Absolutely not. You can see there's all sorts of things going on here in a cell membrane. You have these proteins here that we call carrier proteins. Remember I said some things, small molecules, water, sugar, these kinds of things can easily pass through this membrane. Some other things cannot. Remember those macromolecules, protein. These are huge molecules. They can't get through here. They need help to get through and out. Okay, we'll talk more about this when we talk about cell movement there. Okay, there are those microtubules, cytoskeleton there that's holding the cell in place. Beautiful slide. Let's go. Now, the cell membrane, primarily composed of this phospholipid bilayer with protein, okay, provides structural support, binding sites for hormones, and provides channels there so water and dissolved substances can flow in and out of the cell. We call it a semi-permeable membrane, okay? Only some substances there can cross over, okay? And again, the same thing there. There's your glycoproteins there. These are these carrier proteins there that can allow things in and out. Okay, selectively permeable. The nucleus, we might as well just start off there with the brain of the cell. It's the largest of all organelles, contains the genetic library, the blueprint for synthesis, creation of nearly all cellular protein. As I said, we can store sugar as fat. A can of Coca-Cola, 0 fat. Is it 12 or 13 tablespoons of sugar? It's ridiculous. You drink this there, your liver actually fibrillates, okay, because it's a huge amount of sugar, right? We can store that as fat. We can store fat as fat, but protein we can't store, okay? So we need a constant supply of it. And how do we make protein? We'll show you that there later on. We go back to the DNA. We find the genetic code for that particular protein. We make a flimsy paper bag copy of it. This copy travels outside of the nucleus, where then it gets read by a ribosome, and that's how your protein's made. Pretty simple. We'll get there. Responds there to signals there that dictate the kinds and amount of protein that need to be synthesized. So the DNA, the nucleus there houses the DNA, and we go back and we read that DNA if we need a particular protein made, which is all the time. Most cells are uninucleate, okay, only one nucleus, but skeletal muscle and certain bone cells there and some liver cells there, they can be multinucleated, having more than one nucleus. Red blood cells, anucleate, no nucleus. Red blood cells there, after about 3 to 4 months there, they get the chomp, okay? And our feces there tends to be brown because the majority of it there is old RBCs, okay? Or the remnants of old RBCs. The nucleus has 3 main parts. There's the nuclear envelope, nucleoli, and chromatin. And you'll see here, inside the nucleus controls the workings of the entire cell, surrounded by a double-layered nuclear membrane. Cytoplasm is in the cell. Within the nucleus, though, it has its own gel-like matrix. Okay, we call this their nucleoplasm. Okay, there's your nucleolus, involved in synthesis of ribosomes, creation of more protein-making organelles, and your chromatin. Okay, thread-like structures that contain the genes of our DNA. All right. The nuclear envelope, double membrane barrier that encloses the jelly-like fluid, the what we call the nucleoplasm. Outer layer is continuous with rough endoplasmic reticulum. Okay, you remember that? We heard that word there earlier. So the outer layer is continuous with the rough endoplasmic reticulum. This guy here houses ribosomes, and like the rough ER is studded with these ribosomes there. Those are the protein makers. The inner layer, called the nuclear lamella, is a network mesh of proteins there that maintains the nuclear shape, okay, and acts as a scaffolding there for the DNA. Nuclear pores. There are holes within here, and you'll see them there. You'll see these nuclear pores, okay, because if we need to get into the DNA and make a copy of it, well, that copy has got to get out to the ribosomes that are over here. So there's nuclear pores that are involved, okay? They're guarded there by nuclear pore complexes there, which regulate transport of specific large molecules. The chromatin, welcome to the DNA, consists of 30% thread-like strands of DNA, 60% histone proteins. And you'll see there that your DNA, it's a ridiculously large molecule. And you'll see here how it's wrapped around. You see these proteins there, these circular proteins, they're called histones. That's what keeps the DNA, what's the word I'm looking for? It kind of controls it, conforms it, keeps it in its, that's why it looks this way, right? Because it's so big. And you see, there's those, it kind of looks like a fuzzy sweater, okay? This is the chromatin right here, and you'll see we zoom in, and you just see how massive this molecule is. And these histones there, it's just a way there to maintain the DNA and keep it in a coiled fashion until it needs to be uncoiled. Ranged in a fundamental unit there called nucleosomes, which consist of DNA wrapped around these histones. Okay, chemical alterations of the histones have an effect on DNA and therefore can regulate gene expression. When we're talking about regulating gene expression, okay, here we're talking about controlling your genes. Okay, and your genes, I'm not talking G-E-A-N, what you wear on your legs. Genes here, we're talking about sequences of DNA that, if read in a particular order, will provide us the information to make things. When I mean things, I'm talking proteins. Okay, so the chromosomes there are condensed chromatin. Condensed state helps protect the fragile chromatin threads during cell division. Okay. Here is where we are at. These are the organelles there that we'll have a look at there today. Are there more? Sure. When you're talking cell differentiation and specialization, there are other aspects to different cells, but generally speaking, all of these here are good to go. Okay, we've got mitochondria. Okay, this is the powerhouse of the cell. Ribosomes, protein, endoplasmic reticulum houses these ribosomes. Okay, but you can have free ribosomes as well. You've got the Golgi apparatus involved in packaging, lysosomes, cytoskeleton, centrioles, microvilli, cilia, flagella. We'll get there. Let's go. So, starting off with the mitochondria, okay, there's two layers here. There's a smooth outer layer, okay, and an inner layer folds, and you'll see these folds here. These are called cristae, okay, these folds in here. The mitochondria are tiny slipper-shaped organelles that are the power plants of the cell. They produce most of the energy, ATP. That is our energy currency, okay? Without eating any food, we're only left with ADP, adenosine diphosphate. But once we eat a banana and we're able to extract that energy from it, that energy goes into an ATP molecule, adenosine triphosphate, and now this thing is it's ready to go to do muscle movement, okay? The number of mitochondria per cell varies because of the metabolic activity of a particular cell. Some cells are more metabolically active than others. The harder a cell works, the greater number of mitochondria, okay? Just another shot there of the mitochondria, okay? We've got a real electron micrograph of what's going on here. And you'll see there's the gel matrix there, and as well you'll see these little cristae, these little folds in here. And that's where the energy from the banana that we eat gets transformed into ATP. Okay? Good. Ribosomes. These are the site of protein synthesis, as I've said a couple times there. You're going to hear me harp about ribosomes. They are crazy important. Protein, 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 right? There are fixed, okay, which we're talking about the synthesis of exportable proteins. So maybe the cell makes a protein that exports it to other cells. Love it. There are free ribosomes that are just kind of floating around the cytoplasm there. These ones here synthesize proteins that are used within the cell. All right. So you have fixed ribosomes and free. Endoplasmic reticulum. There's two kinds. There is rough and smooth. Okay? Rough endoplasmic reticulum, what we call RER, rough endoplasmic reticulum. This one here houses these fixed ribosomes that are not free to move. Okay? For exporting a protein. Maybe the cell is making a protein there that the body needs, and those proteins have to get out of the inside of the cell and to the outside. And those, ow, nice. So these fixed ribosomes there, they export the proteins out, and the free ribosomes, they're for proteins that are used intracellularly, if that's even a word. Smooth ER, okay, there's no ribosomes. Here is the site of lipid and steroid synthesis, okay. If I'm going to know one of these, a little bit of advice, it's that one. Okay, ribosomes are important, and rough ER. Okay. Golgi apparatus here, we're talking about packaging, packaging, packaging. Now, you remember there, that semi-permeable membrane, okay, that phospholipid bilayer, okay, of the cell, the outside of the cell. It's selectively permeable. Some things can get in, some things can't. And those things that can't get in or out, they need help in getting in or out of the cell. So what happens here, like let's say the body, the cell is making a protein. It can't get outside of the membrane to get to where it needs to go. So what we do is we package it with a little bit of the membrane. Okay? And just going back into, where did we go? What slide is that? I don't want to lose that. Okay, it's 20. Yeah, right in here. You'll see here that these are proteins, and they can't get outside the phospholipid bilayer by themselves. So what the Golgi apparatus does is very closely situated there with the rough ER, okay? And these proteins are made by the rough ER, and then they're immediately packaged by the Golgi apparatus, which creates these little bubbles, which are exactly the same composition as the cell membrane. So it floats over, and you'll see here there that these little packages, they open up when they hit and the protein is allowed to get out. And this just ends up becoming this package ends up becoming just part of the cell membrane. Okay, so that's your Golgi apparatus. And where was that? Number 20. All right, so packaged proteins in the Golgi membrane there puts finishing touches on protein after synthesis in the rough ER. And there's a beautiful electron micrograph of a Golgi apparatus. Okay, lysosomes. Okay, these are the housekeepers of the cell. Little bits and bobs of protein, fat, this and that get left behind. The body's perfect as best as it can be, but it's not 100%. So here we have these lysosomes there that go and clean up and break down, okay, waste. The cytoskeleton we already talked about. These are the microfilaments and microtubules there. They maintain the shape of the cell. And then we have centrioles there. These are rod-shaped structures. They help separate chromosomes during mitosis. Okay. Microvilli. You see these here on some type of epithelial cells, especially a good one there just off the top of my head in the intestinal tract. Okay. The whole idea there, the body is a marvel of technology, and most specifically at creating space where there is no space. You know, like when you're talking your lungs, you know, you're breathing in oxygen, and it's all about surface area. If you have surface area for your lungs, then the oxygen comes in, hits the wall of the lung, alveoli, which we'll get there, and the oxygen moves over to the other side where the blood vessels are. So that's how the oxygen gets into your blood. The carbon dioxide, which is on the other side, okay, it permeates through in the inside of the lung and is able to be expelled. It's all about surface area. And the lungs there, if you spread them out, all this surface area has the same surface area as a tennis court. Your small intestine, how is something there, you know, your duodenum, okay, how is it able there to absorb all of the nutrients there from your Big Mac combo? Well, again, something there it's a relatively small space. Your small intestine, not big at all. But again, it's all about surface area. And you see what's going on here. You have these cells, but then you see what the body does is you have these little projections coming off of these cells. And it's all about increasing surface area. It's surface area, surface area, surface area. We'll get more into these there when we get into the digestive system there. So those are microvilli. Cilia. These are short hair-like projections. You'll see there, if you're talking there, the respiratory system inside the lungs. We produce mucus. Mucus is one of those primary defense systems there that we have, okay? Because we breathe in 500,000 bacteria with every single breath. So we're constantly breathing in bacteria. And we don't want these in our lungs, but we know that they're there. And you see our lungs there produce mucus. And these bacteria come in with the intent of harming us. And all of a sudden there they get trapped in the mucus. And now we don't want that mucus in our lungs. So what happens there is these cilia, okay, they kind of work like seaweed. You know, you'll see these seaweed there at the bottom of the ocean there with the waves, right? And that's what these guys do. They push this mucus up, 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 until eventually there we're able to expectorate there, and that's what it is. Yeah, that's gross. We're going to get a lot grosser. All right, that's the body. Okay, flagella there. Here we're talking the tail. Okay, some bacteria have flagella. Okay, for motility. Sperm cell here enables it there to move and navigate to where it needs to go. We'll cover that one there in more detail for sure. What's going on here? There we go. That's what I wanted. Centriole there forms the basis of the cilia and flagella there, and we'll get more into that. Movement in and out of the cell. This is pretty important stuff. The whole idea there that why does a ball roll down a hill? Well, the ball rolls down a hill because somebody pushed it all the way up. Somebody put the energy into it, and then it rolls down. Is that always the case? Well, no. You see here, sometimes we need to input some energy in order for things to move. And you remember there, the whole idea of diffusion. Things generally move naturally. Doesn't cost us any money. And when I mean money, I mean ATP. Doesn't cost us any money, okay, when we're talking high concentration to a low. Remember that? And then in 5 minutes, high concentration, low concentration, and generally there, things will form an equilibrium and eventually balance out. All right. Sometimes there, we have passive diffusion when we're moving things across a membrane. Free money, doesn't cost us anything. But sometimes there we have to move things from a low concentration to a high concentration, which is not natural. So therefore, in these particular cases, it does cost us money, ATP. When we're moving things from a low to a high, costs us ATP. When we're moving things from a high concentration to a low concentration, generally free money. And that's how the body does this. And we'll show you this. The body is amazing at saving money. Transport mechanisms here. There's your passive mechanism. This is free ATP. Doesn't cost us any money. We're going to look at diffusion, facilitated diffusion, osmosis, tonicity, filtration. These are all free. Don't cost us anything. Active mechanisms of transport across a membrane. Yeah, there's a couple of them here where we're moving things from low to a high concentration, which again is not natural. We've got active transport pumps. We've got endocytosis, exocytosis. You've got transcytosis. You've got all these ones here. So, passive diffusion. Okay, passive transport diffusion. Now again, I want you guys to have the frame of mind of moving things from here. It's all about the plasma membrane. Moving things from one side of the membrane to the other, okay, or this side to that side. How do things get there? And the easiest and the freest of all the types of transport is just general diffusion, simple diffusion. Movement of a substance from an area of high concentration to an area of low concentration. And you'll see there, you know those vitamin C, what do they call them, effervescent tablets there? You put it into a glass of water, it starts to fizz, right? And eventually there, after a couple minutes, all of that vitamin C tablet is now diffused throughout, right? There's an equilibrium, okay, the point at which the concentration is equal and no further net diffusion will occur. But this is movement of a substance from an area of high to a low concentration. High concentration right in here, but eventually there you'll see there is an equilibrium and everything gets spaced out accordingly. Okay, this is a great example here. The whole idea is we can smell a skunk, okay, from, after it's sprayed. All right. Now, right at ground zero, I don't know if anyone here has been sprayed with a skunk. I came about this close to being sprayed by a skunk. I mean hit, like from a meter away. And I smelt it and I ran. But a skunk at ground zero smells very different from a skunk from 2 kilometers away. All right. And it's all about very highly concentrated, but then it diffuses out over hours, and that's why we can smell it. Okay, kilometers away. Passive diffusion. Okay, this is what we're looking at right here. See, we breathe in the oxygen. Okay, these are our lungs here. This is our left lung and our right lung. Notice there that the number of lobes of the lung are different from the left and right. And the oxygen comes in through our bronchs and eventually hits our alveoli. These are the end terminals. This is where the oxygen has got to go in order to be diffused into our blood. So this is called an alveoli. And you remember there on the other side of the alveoli, there are the blood vessels. And they are right there. Okay, oxygen comes in, and you see there that the oxygen concentration is here is the same as in our atmosphere, which is 21%. It's pretty high concentration of oxygen when it comes in, the air there. Okay, 21% oxygen. And you'll see there that the oxygen returning to our heart and our lungs there is very low in oxygen, right? That's the whole point of the circulatory system. We breathe in, okay, our oxygen, the oxygen jumps to our blood, the blood then takes that oxygen to our big toe, drops the oxygen off in the big toe, picks up the carbon dioxide, and then returns back to the heart. So the blood that returns back to the heart is high in CO2, carbon dioxide, and low in oxygen. We breathe in, okay, and then the blood going back to our big toe is high in oxygen and low in CO2. That's the whole mechanism, okay? And you'll see here, there's your alveoli, moving this around here, okay? There's your oxygen and moves across, CO2 jumps, okay? Because the CO2 in the blood is higher than it is in the atmosphere, so the CO2 jumps. Didn't cost me any money to do that. And the oxygen there comes in, high concentration, low concentration here, jumps. Okay. Another type of diffusion, free money, okay, is facilitated diffusion. This is where we use a helper molecule within the membrane. Now you remember that slide 27, right in here. Remember these guys here? These guys here, some of them are involved. There are proteins embedded within the cell membrane. These guys here, we call them carrier proteins. And they help those big wonky molecules to get through. But again, it's all high concentration to a low concentration. Free money. That's what we call facilitated diffusion, where a helper molecule within the membrane assists with the movement of the substances from a high to a low. Free money. Osmosis. Now this one here is a little different. Here, when we're talking about facilitated diffusion and simple diffusion, here we're talking about a particle. Something that is moving from one side of the membrane to the other. Osmosis, we're not talking about a particle. Osmosis, we're talking about water only. Okay? And you'll see there, it's the movement of water, okay, from an area with more water to an area with less water. So here we're talking about concentration again, free money. I don't need to input any ATP in order for this to happen. Things move from a high to a low concentration naturally. Free money. So you'll see here, so here I have a beaker. I put in what we call a permeable membrane, selectively permeable membrane. Okay, some things can get through, some things cannot. So what we have here, we've created a dual system. So here I have one solution, okay, tends to be pretty dilute. You can see the particles are kind of spaced out, okay, and on this side of the membrane there, I have the same particle and then it's a higher concentration. But this membrane will not let these guys travel through. It will only allow water to travel through. So if these guys here, these particles here, they want to get through because, you know, they don't like each other. They're in close proximity to each other than closer proximity to each other than these guys here. They want to get through, but they can't. So what will happen is the water will move from a higher here we're talking water. The water here has a higher concentration per particle than these guys here. So what happens there is this water will move through. And you'll see there, it will form an equilibrium. And you'll see there that the water will travel through until everything gets spaced out across both. And you'll see there that the water will rise. It looks strange, doesn't it? But it does. Okay? So that's what we call osmosis, the movement of water from a high concentration to a low. And this is pretty important stuff. And you'll see there the ability of a solution there to affect the volume and its pressure within a cell. We call this tonicity. Okay? You can have an isotonic solution, a hypotonic solution, and a hypertonic. Now you think hypothermia, hypothermia, low, hypo, low, hyper, high. Okay? So you'll see here that if we take a red blood cell, and our blood plasma and everything, the pH and all the electrolytes there, it's there for a reason, and we'll get more into blood there later. Okay, but if I take an RBC from my blood cell and put it into a solution that has the same salt concentration, remember those ions, as my blood, you'll see there that water entering the cell will be the same as water leaving the cell. So this is a happy RBC. Okay, it's what we call an isotonic solution, where it has the same concentration of ions as the original solution. So this cell here looks great. But now a hypotonic, low salt. Okay, this is what we're talking about here, and you'll see there that the water concentration is greater outside than inside. Okay, and what will happen there is the water will come into the cell, and the cell will what we call lyse. It'll blow up, okay, because the salt concentration inside the cell is greater than here, so the water will move inside and blow it up. Now, if I put that RBC into a salty solution, okay, now the water concentration inside is greater than the water concentration outside, so the water will leave the cell and it'll turn into like a prune or a raisin. Okay, so you have isotonic, same salt concentration. Hypotonic has a lower salt concentration than the RBC, and then a hypertonic, okay, where the salt concentration in the solution is higher than inside. So this turns the RBC into a raisin. All right. Another type of free money transport, when we're, again, remember, we are talking about movement from one side of the cell to another, either inside to outside or outside to inside. Here we're talking about free money. There's another way that things can move freely. Doesn't cost us anything. It's called filtration. And you'll see here, if you look at a hypodermic needle, if I want this stuff to get, whatever this medicine here, I want to inject it into the patient or whatever. Here we're talking water, whatever. And if I want to get this water out, I have to put pressure on here. And increase pressure, now I'm decreasing the volume and this stuff shoots out. If I want to bring the medicine in, when they put the hypodermic needle into the medicine there and then they pull on the plunger, okay? This is, here we're moving things around. So the movement of water and dissolved substances from an area of high pressure to an area of low pressure. Okay, so you increase the pressure on one end, it comes out. And you see the force of the heart. Here we're talking a blood vessel. And these blood vessels are not just like Super Mario pipes, where they're just solid. You'll see there that there's what we call fenestrations. And there are actually kind of areas within the blood vessel that will allow these RBCs, phagocytes, all these kinds of things to get out whenever they want. So the force comes from the heart. That's the pressure. Okay? And this pressure here will allow these things to migrate out and get to the interstitial fluid where they need to be. So water and dissolved substances are pushed out. So there's filtration, diffusion, facilitated diffusion, and osmosis. That's all free money. Moving on into active transport. Okay. Now this one, not diffusion. Diffusion was free money, high concentration to a low. Okay. Sometimes though, we need to maintain an imbalance in the membrane. We'll get more into this there when we get into nervous system. We get into lots of other stuff there. But you'll see there that sometimes we have to move things against the concentration gradient. So this is the movement of substances from an area of low concentration to an area of high concentration. okay? Which seems weird because naturally things go from a high to a low, but sometimes in the body there we need to maintain and we need to move things from a low to a high, all right? This one here requires ATP. This one here cannot be done unless we have energy from food, okay? So active transport here, endocytosis. Endo means entering. So the ingestion of a substance by the cell membrane. Maybe there is some protein outside of the cell and it wants to get in. Well, this protein is floating around the blood here, and the body and the cell wants it inside the cell. So what happens there? The cell membrane will invaginate. There will be this hole created. The protein there is brought in, and it's encapsulated. Okay, it's kind of like the opposite of the Golgi apparatus, right? It's making packages to send out. Well, here, this whole process of endocytosis is bringing something in, and the cell membrane encapsulates, and now we're able to bring that into the cytoplasm to do what we need it to do. Okay, there is phagocytosis. That's the Pac-Man. Okay, if there's a bacteria there that is causing us trouble, well then what happens? The chomp chomp, it engulfs that bacteria and breaks it down. Pinocytosis, here we're talking about a liquid. If a liquid there, if the cell needs some water or some type of liquid there within the plasma stream, okay, we can bring this liquid in through what we call pinocytosis. Phagocytosis is the ingestion of a solid particle. Pinocytosis is the ingestion of a liquid, bringing it into the cell. There is exocytosis. We saw that already there with the Golgi apparatus. The cell makes a protein and wants to export it out, while the Golgi apparatus there packages it up. This guy here travels to the membrane, and you'll see that this packaging actually becomes part of the cell membrane, and these get released out. Okay, so exocytosis, releasing something to the outside extracellular matrix, outside the cell. Endocytosis, bringing it in. Cell division. Now we're going to tread very lightly on this. Okay, when we're talking mitosis, it can be quite complicated, and it's not a focus of this course at all. As long as you're aware that cells, there's two ways there that cells will divide. Our body cells, okay, these guys here, they replicate through mitosis, where you get one cell, okay, that will double, its DNA will double, and you will end up with two identical cells of that one. Welcome to your body cells, what we call somatic cells. That's how we grow. That's how we maintain. Okay? But then there's another one there called meiosis. Okay? That one's completely different. Here we're talking about sex cells. Okay? Sperm and egg cells. And I don't want to get into it too heavy right now, but these sex cells, every one of our sex cells there, sperm and egg, have never, ever, ever, ever been seen before in the known universe, which is the same reason why a mom and a dad can have 4 children. The same mom, same dad can have 4 children that look nothing like each other, because we don't want people to be clones. Now you think, why? That'd be a heck of a lot easier, right? If I had 4 kids there and they were all the same, bang, 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 not a problem. The reason why we don't want our sex cells to be the same, because if there is a disease that is out there, and we just went through a pandemic, if there's a disease that comes, and we know there's going to be more pandemics, the disease, bacteria, viruses, fungi, they were here billions of years before us, they'll be here billions of years after we're long gone. Okay, the whole idea there that if there is a disease that wipes out one person, if all the children are clones, then you can just say goodbye to your species as a whole, right? It makes sense, right? If a disease is going to take out one person, okay, we've lost one person there, but the 3 others there, they will grow and have offspring, and our species will continue. We'll get more into that as we go. But there's your mitosis, body growth and repair, which tend to slow down as we get older. There is meiosis. This is sex cells only. Okay. The cell cycle, the sequence of events that a cell goes through from one mitotic division to the next. And again, I'm treading very lightly on mitosis and meiosis at this point in time. Okay. Cells have a limited lifespan. Okay. So cellular division is necessary for body growth and repair. Even cells get old. And old cells that don't divide anymore, guess what happens to them? Chomp, chomp. Some cells, such as skin cells, bone marrow cells, and the cells lining the digestive tract, reproduce very rapidly. Our skin cells, every 4 weeks or so, we have new skin. The whole idea there that when you're sweeping up dust in the kitchen floor or wherever you're sweeping up dust, know that the majority of that dust is skin cells. Yeah, this one here is going to gross you out even further. You buy a brand new mattress, you know what I mean, and you're carrying it up the stairs, and what does this thing weigh? 50, 60 pounds, it's all floppy and hard to, but you get it up there and you get it on the box spring, away you go. After sleeping on that mattress for 15 years, that mattress now weighs 200 pounds. Why? Skin cells. Yuck. So other cells such as nerve cells, they have a tough time reproducing at all. Okay, which is why, if you have some type of spinal damage, a paraplegic, quadriplegic there, the nerve cells have a tough time regenerating. Okay. This is the cell cycle. There's generally 2 phases there. You have interphase, which covers all of this. Okay, which is encompassing there the G1 phase, the S phase there. This is the synthesis of new DNA. You have a G2 phase there. And then you have the actual mitosis itself. Okay, and there's prophase, metaphase, anaphase, telophase. Again, I don't want to go too crazy and too heavy into this stuff, but just the whole idea there that if you're familiar with mitosis as cell division for body cells, I like that. And then you have meiosis, which we will get into later on, which is the creation of sperm and egg cells. Okay. The point of mitosis, this one right here, is to create 2 exact copies of the original cell. Meiosis, no. Every cell is different. Okay. So your mitosis there, there's your prophase, metaphase, anaphase, telophase. And the whole point there is the cell are duplicates. So there's 4 phases there, and I call this PMAT. P-M-A-T. All right, but this is an open book test, so you don't have to remember. You can just go to the slides. And that's the nice thing about these tests, is that the tests are going to be stress. I get it. All tests are stress, but you have all the time there that you need. Okay, you can sit there with a cup of coffee, dig through the book. Be careful of Google. Google, you know what I mean? If you Google kidneys, there's a whole, you're going to get all these things there that have nothing to do with Google. So just be careful. If you're going to Google answers, make sure that you choose the right answer. All right? The final stage there of mitosis is what we call cytokinesis. This is the whole idea there of these cells pinching and creating 2 separate cells. You can see here it's happening. Okay. The getting into nerve, you remember nerve cells there, they have a tough time of regenerating. They just really don't. And whether you agree with it or not, there are ethical concerns, but where there's this right here, it appears to be revolutionizing genetic research and cell regeneration. These are stem cells, and these are cells you take there from the amniotic fluid. These are cells there that can go. We can turn them into anything. Are you kidding me? Well, that's what the body's been doing for billions of years. These stem cells there, if the body needs nerve, they can make nerve. They can transform into nerve. Stem cells can transform into, there's your specialized cells, neurons, RBCs, muscle cell, bone epithelial, okay, and all they need is this particular enzyme to do so. So stem cell research, it's the forefront. There are ethical concerns with it, but if you have Parkinson's or some type of nerve damage, there is hope. There really is hope. Relatively undifferentiated, unspecialized cells whose only function is the production of additional unspecialized cells. The rate of stem cell division varies within any particular tissue type. The stem cell within the bone marrow and skin are capable of dividing more than once a day. Stem cells in adult cartilage may remain inactive for years. And this might sound like a strange question, but which is worse, breaking a bone or tearing cartilage? Cartilage there, your nose, your ear, but you also have cartilage there on the ends of all your bones, right? It's the soft, squishy cushion, okay, that protects bone against another bone, right? Because you never want bone touching bone. Ugh, just makes my skin crawl just even thinking about it. You want the cartilage on the bone to touch the cartilage, soft, spongy, touching soft, spongy. What is worse, breaking a bone or tearing cartilage? Well, bones make blood. They got lots of blood. So it's a quick, easy fix there in, 4, 5, 6 weeks. Cartilage, no. Cartilage has a very, very low blood supply. So if you tear cartilage, it takes considerably longer to heal that than even a bone break. Okay. What do we got here? And last slide for chapter 3. Some disorders there of cellular growth. You've got abnormal cell growth. These are tumors. Now, there are tumors that are benign, non-cancerous, like a mole. Okay? These guys here. The bad one there that you never want to have is what we call a malignant tumor. And this is cancerous. This thing is gung-ho. This thing is more than alive. It's aggressive. It's eating nutrients, absorbing oxygen more so than other cells. So these surrounding cells, which are yours, the cancer is not yours. It steals a lot of the nutrients around, and these cells starve. These cells extend and invade surrounding tissue, taking resources. And 2 words there I'd like you to know. There's necrosis, death of cells, and there is apoptosis. This is also the death of cells, but it is the programmed death. Okay? And we'll get more into that there, but some cells, they're designed to die. Okay? And they help rid the body of old and unnecessary and unhealthy cells there. We'll get more into this there as we go. Thank you so much for watching here. Let's take a 10-minute break. I'll see you guys back here at 10:30, and we'll continue on with chapter 4. Okay, does anyone here have any questions? How's everyone doing? Is there anyone still? I see that there's lots of people still here. I'm joking. But if you guys have any questions, let's chat with them there at 10:30, and we'll see you guys. Go get a coffee, and we'll see you back. Thank you so much.